parallel with petunias. In the field, they tested a method that prevents the spread of newly introduced genes. The genetic information is not inserted into the cell nucleus, but into the plastids. Plastids are small subunits within plant cells that possess their own DNA. Since the genetic information of the plastids is not passed on via pollen, the DNA remains biologically confined in the plant. If genes are in the plastids and not in the cell nucleus, the probability of them being transferred to seeds of other plants via pollen is smaller. This would mean that the coexistence of genetically modified and conventional plants would become much easier. By now, we have greenhouse trials which indicate that for the majority of cases, the confinement works. However, sometimes a gene transfer occurs, and therefore we want to find out in the field whether we get any gene transfer, and if that is the case, how frequently this happens. Genetically modified petunias with purple blossoms and non-modified petunias with white blossoms were cultivated on 3 large trial areas. The pollen of the genetically modified petunias was supposed to pollinate the white non-modified petunia blossoms. Around each row of genetically modified petunias, we planted rows of non-modified ones. And we then either waited for them to be naturally pollinated, or for most plants, we actually took pollen and applied it to the pistils of the wild-type plants in order to increase the rate of cross-fertilization by the genetically modified pollen. The seed capsules were harvested and the seeds were left to germinate in the lab. A special gene was introduced into the genetically modified petunias, which causes a blue staining of the sprouts. Therefore, the sprouts were searched to see which of them carried the blue color. The ones that did were tested whether the color was really caused by the new genetic information. In each year we analyzed 30,000 seeds. We let all of them germinate and tested them, and of course we also had to test whether they were actually cross-pollinated ones or self-pollinated. Although we virtually excluded self-pollination by removing the anthers and applying the pollen of the modified plants to the pistils, we only found very few cross-pollinated ones. Not a single one of them in the field contained the blue staining via a pollen transfer. For this plant, we're able to say that such a process will not occur, but we're not able to generalize the result. For other plants with a higher rate of cross-pollination, one will have to test this again. What this experiment nicely demonstrates is that we cannot directly compare different growing conditions. The data from the greenhouse were very clear and cross-pollination worked excellently.